In the context of the Biodiversa Plus project called ProPartS, this video shows information about our studies on the niche dimensions and the restoration potential of the charophyte Chara canescens. Apart from knowing that Chara canescens lives in brackish water, the exact habitat requirements of the species are not precisely defined, and conditions vary from location to location. Therefore, a series of ongoing analyses are being performed to find out more about the sediments in terms of their physical and chemical properties, the sediment microbiome, the water chemistry, for example nutrient concentrations, or presence of other vegetation. Because the microbiome plays an important role in the ecology of plants and algae, the taxonomic composition of microbial and fungal communities is determined in the bulk soil around the rhizoids, the roots of charophytes, using a next-generation sequencing approach. Bacteria and fungi from the endosphere and rhizoplane of the rhizoids are cultivated on agar plates, and the resulting isolates are identified taxonomically using Sanger sequencing. The main propagules of charophytes, called oospores, are preserved in the sediments, and they are the source of new generations from their germination. The study of their morphological characteristics also provides interesting information. Sediments from all sites are analysed to determine, in different layers, more superficial, then more recent, and deeper, then older, how many oospores they contain, and whether these oospores are viable or not. Moreover, we perform germination experiments which allow to confirm the viability of these oospores buried in the sediments and calculate germination rates. This informs us about the restoration potential of, for example, dried water bodies. In our other videos, you can get detailed information on the different aims of the project, and remember to visit our webpage for updated information.